Evolution implies change, and it is the process by which we track changes that occur through time. Many believe the concept of evolution to be contentious, but it shouldn't be. All things change, and evolution is just a record of those changes through time. Charles Darwin is considered the father of evolution. His groundbreaking work began when he spent 5 years aboard the HMS Beagle. He was hired as an educated companion and naturalist for the captain of this small ship. During this time, he sailed a route that was literally around the world. He collected and cataloged thousands of plants and animals and made numerous observations, took copious notes on species variations and their relationship to fossil forms. From Darwin, we have a better understanding of biodiversity, the fossil record, and what he coined as descent with modification, the passing of traits from parent to child with subtle variations. Before Darwin, the accepted age of the Earth was determined by James Ussher and John Lightfoot, who made the assumption that the Bible was the only reliable source of chronological information, and therefore calculated that the Earth was created on Sunday, October 24th, 4004 BC. They even put the time at 9 a.m. A century later, Comte Buffon believed he could get a better estimate of the age of Earth based upon its rate of heat loss. With his mathematical equation, he calculated the age of Earth as 74,000 years old. But Darwin, due to an earthquake that uncovered fossils even older than 75,000 years, demonstrated that the Earth had to be much older than previously thought. And he made the connection that these fossils were representatives of previous forms of life, past examples, slightly different, perhaps more simplistic, but that they were interrelated. This provided a framework for the way we study and understand the living world. Layers of rock can accurately be dated by their strata, and comparisons of bones can reveal alterations over time. This creates an orderly sequence of change and replacement of organism and life that we can track over billions of years, not just thousands or tens of thousands like the early estimates occurred. We can also see the interconnectedness of life by looking at homologous structures, examples of organs or bones that appear repeatedly in different animals, underlining anatomical commonalities, likely singular origins that were passed down from an early time point in life. If we look at global patterns and biogeography, we can see that shared patterns of organism life are consistent with the occurrence of Pangaea, one large global continent that were once all connected. And over time, when that broke apart, this created new pressures and new speciation events, consistent with those tectonic movements. With modern tools, we can expand to include molecular biology. We know that DNA is a universal molecule for all living organisms, and it has a near universal genetic code. 90% of life begins with AUG as a start codon. And by comparing sequences by sequences, we can track the accumulation of mutations, copy by copy, organism by organism, further supporting the interrelatedness, as well as phylogenetic trees showing these divergent lines and descent with modifications. Macroevolution really focuses on differences that beget a new species. Speciation is defined as the origin of a new type of organism, a distinct form. In order to validate a new species, we must be able to track where it came from and explain the how it evolved, what makes it different, and what pressures led to this. Throughout science, there are many different species criterions used. A few are biology, morphology, ecological niche, and phylogenetics. The biological species concept looks only to determine whether or not they can mate and produce viable young. And so based upon the biological species concept, we can see a wide variety of human variation, but one singular species, because gene flow between populations maintains this diverse phenotype. Meanwhile, the morphological species concept defines a species by distinct structural features, having a different color or a different size beak. The ecological species concept views a species in terms of the ecological niche. A niche is both where it lives as well as the unique role it plays within that ecosystem. So here we see the African forest and the African savanna elephant. They are two distinct species, similar in appearance, but because they vary in where they live, what they eat, and how they interact with the environment, they are distinct forms. The phylogenetic species concept defines a species as the smallest group of individuals on a phylogenetic tree. So it's looking at the shared family history. Another species concept is the genetic species concept, looking at specific nucleotide differences. There's also a behavioral species concept, which defines species based upon distinct patterns of seeking or showcasing for a mate. The eastern and western meadowlarks sing different songs to attract their mates. So even though they can coincide in the same environment, they are never attracted to the opposite species. It's just not their type of music. There are a number of pressures that will elicit change. Mutations occur randomly. Genetic drift or sexual preference have impacts on who's mating and procreating. Environmental shifts or barriers can ultimately affect who lives to procreate. All of these things are the groundwork for natural selection to act. Speciation can occur in two ways. Allopatric speciation occurs in geographically distinct separate environments. Sympatric speciation occurs when the organisms are in the same environment and they're changing simultaneously as a means of decreasing competition and specializing further amongst their shared resources. Allopatric speciation often occurs when gene flow between a population is interrupted or reduced. Some sort of barrier has been erected, cutting off gene flow, separating the populations. And because they are separate, they are subjected to different environmental pressures. Separate populations may evolve independently through mutation, natural selection on distinct scenarios, and differences in the proportion of alleles on either side of this divide. It's important to point out that what may be seen as a barrier for one organism may not be a barrier for another. Sympatric speciation, as mentioned before, takes place in an overlapping population. This type of speciation is much more complicated. It may include polyploidy, extra genetic information from hybrids that are starting to mate and intermingle, habitat differences, sexual selection, ultimately outside pressures such as competition driving them to become unique. Oftentimes, when an organism is evolving, we see what is known as adaptive radiation, the diversification of an entire group filling different ecological niches. Evolution doesn't necessarily occur in a single path to make things better. The accumulation of mutations creates several scenarios, and as those continue to be supported through natural selection, we see more and more organisms following in suit. Darwin's finches, for example, were 14 different species in the Galapagos Islands, and each finch, in order to reduce competition, started to specialize in what they ate. And it's not a choice. The finch didn't choose to eat cacti versus bugs versus seeds. It was that random mutations affected beak size. And depending upon your beak size, well, form dictates function. That determined what type of food you could easily obtain. Darwin saw his finches diversify rapidly from a single ancestral species into a multitude of new forms. This was the result of rapid changes in the environment, creating new challenges or opening new environmental niches. While all traits are considered equal, oftentimes biologists consider reproductive isolation the end all be all dividing line of divergence. To be reproductively isolated means that there are biological factors, some sort of barrier that will stop two species from mating and producing a healthy, viable, and fertile offspring. These barriers can be further classified based upon whether or not they act before or after fertilization. Prezygotic barriers occur before fertilization. They stop sperm and egg from joining. A prezygotic barrier may stop them from even attempting to mate, from completing the act of copulation, or from the actual union of sperm and egg. And so there are many forms that these prezygotic barriers can take. We can separate a mating pair by habitat. Perhaps they never encounter each other. We can separate them based upon time. Perhaps their interest occurs at opposite intervals. We can separate species based upon behavior. What if they don't recognize the signs and signals that the other is putting out? There can be mechanical barriers having to do with the actual genitalia of the organism, and gametic barriers referring to incompatibility of the sperm and egg. And so this diagram has examples of each of these types of prezygotic barriers. Habitat isolation, these are two types of garter snakes, one that mates in the water, one that mates on land. Even though they technically could mate, they won't ever because they're looking for love in all the wrong places. Temporal isolation is examining two different skunks, one that breeds in the spring and one that breeds in the fall. Even where they co-occur, they won't mate with each other because they just can't seem to get their timing right. For behavioral isolation, we're looking at a blue-footed booby. This bird depicts its interest in a mate by tap dancing and flashing their brightly colored feet. If you weren't a blue-footed booby and attracted to bright colored feet work, you'd be looking at this bird going, that doesn't look right. And so that can be a turnoff. With mechanical isolation, it's about fit. Does it work? With gametic isolation, the example here is sea urchins. To prevent other species from mating with their own, they actually secrete their own inhibitors and damaging enzymes that would kill off the sperm of nearby sea urchins, and therefore only their sperm would be able to swim in the surrounding waters and be available for fertilization. Postzygotic barriers, on the other hand, prevent the hybrid zygote from developing into a living and fertile adult. Postzygotic barriers may take the form of reduced hybrid viability, meaning conception occurs, but the fetus doesn't develop as robustly, resulting in stillborn or early infant death. There's also reduced hybrid fertility, resulting in a sterile offspring, or hybrid breakdown, which is seen over multiple generations, with each generation becoming more and more feeble or sterile. Here we can see three examples. Newts and salamanders can mate together and achieve fertilization. However, no live birth result from this union. When donkeys and horses are mated together, we get a mule, a robust organism on its own, but unable to reproduce. And in the last panel, we have an example of some wheat strains that are depicting hybrid breakdown. And we can see the multiple generations get a little smaller and a little less robust. This is usually due to an accumulation of recessive alleles. As we expand on our understanding of evolution, we will now discuss the tenets, the rules, and generalizations that have become the foundation of this field. The first is that variations occur. No species or organism is exempt from that. The second is that these variations have a genetic basis, and sometimes they are passed down generation to generation. The third is that a mating pair will produce more offspring than will actually survive to maturity and the ability to reproduce on their own. If not, one bacterial cell in 36 hours could cover the Earth 3 to 4 feet deep. The fourth touches upon fitness. The variations that best fit the environment are more likely to survive and reproduce on their own. Those with less favorable variations will be less likely to survive. In this struggle for existence, natural selection will sort through the different variations and choose the most fit combination. In the diagram here, we've got giraffes of variable length, and we can see that the ones with longer lengths were able to reach the leaves and eat, and therefore live through to reproduce, while the one with the stumpiest neck died from starvation and took those short, stumpy genes with them. And the sixth and final tenet is that because of these variations and the force upon which natural selection plays, populations will be able to adapt gradually to changing environments. How can we tell where we are in the process of evolution? How can we tell how much change has occurred and quantify how much change there is yet to do? When we look at the historic record based upon fossils, two models have emerged. The first is known as gradual change. This is what Darwin described as descent with modification, that each generation, small variations, small changes would eventually lead up to big speciation events. The second model, known as punctuated equilibria, describes the occurrence of a sudden and brand new species that acquired most of its defining features at the moment of its inception, immediately. It is hard to define a species because there are many species criterions and they are not inherited in any particular order. Sometimes they may be inherited at the same time. Sometimes it takes decades and generations for the next feature. And sometimes the species criterions may be contradictory, as in this example here. We're looking at a Y showing a single species at the base and two separate species at the tips. And both examples, the dogs and the butterflies, are depicting the morphological species criterion. On the left, based just upon morphology, we would say these dogs were two very separate species. They don't look alike. And yet we know from genetics, biological species concepts, ecological species concepts, and phylogenetic species concepts that they are actually one. On the right, with the butterflies, based on morphology, we would place these two together as a single species. However, based upon biology, genetics, ecology, we know these two to be two separate species, the monarch versus the viceroy. And so herein highlights an area where we're unsure, based upon which species criterion you're using, if we're really discussing one species or two species. This gray zone highlights an area and the occurrence of cryptic species, species that are actually different from one another, but go unnoticed because they are not morphologically distinguishable. The example below are an assortment of butterflies that actually represent 8 different species, 4 different genera, and 2 subfamilies, meaning that they are genetically very distinct. And yet at first glance, we might only see 2 variations here. To help us discover new species, we are relying more and more on DNA barcoding and the genetic sequences of each organism. To complicate things further, I want to highlight and remind everyone about convergent evolution, that sometimes traits can be shared without a common ancestor. We use the term convergent because it depicts a scenario in which 2 separate lines get to the same place. They converge upon the same instance, the same phenotype. They appear to be similar because evolutionarily, natural selection has chosen these traits to be the most fit, but not because they're related. Another complication is the fact that sometimes separate species can continue to hybridize, breed, and form viable young. The hybrids are the offspring of these crosses. They may look like either parent or an intermediate form. Sometimes those intermediate forms can vary depending upon who's the male parent. Tigons are produced when a male tiger breeds with a female lion. Meanwhile, ligers are the offspring of a male lion bred with a female tiger. In this instance, the specific parentage differentiates their phenotypic presentation. Ideally, when we cross species, we're aiming for hybrid vigor when the offspring is better than either parent. In an area where there's constant crossbreeding and hybridization, we can form a hybrid zone, a single band where these adjacent species meet and commonly procreate. In this region, you may see 3 different phenotypes, both parental species and the hybrid. A hybrid zone has added selective pressure, which can result in 3 patterns depending upon the relative fitness of the hybrid. Be aware of these 3 patterns, be able to recognize them and depict them. The first pattern is known as reinforcement, where the hybrids are less fit than either parent species, and so it reinforces the idea of 2 separate and distinct species. Over time, the rate of hybridization will decrease, and reproductive barriers will evolve to prevent the misallocation of resources in trying to mate. For example, in populations of flycatchers, males are more similar in allopatric speciations than sympatric populations, meaning if they co-occur in the same area, they phonetically look different to prevent mistaking the wrong male and breeding with them. The second pattern is known as fusion, in which case there's substantial gene flow between the parent species and between hybrids and either parents. The hybrids are just as healthy and fit, and so they're equally likely to mate with each other and the original parent populations. Over time, this will make one very diverse species. 2 separate species will fuse into one single species. Such was the case for the Pundamelia species in Lake Victoria. In the beginning, there were 2 very separate species that would not mate with each other due to morphological differences. However, when pollution occurred, it reduced the ability of the females to distinguish the males of different species, and they started reproducing. And because there hadn't been any reinforceable barrier before, in this case, pollution weakened the reproductive barrier of morphology. The final pattern is known as stability. In this case, there's the continued formation of hybrid individuals, but the hybrids do not reproduce with the parents anymore. Over time, this will result in 3 distinct species. The overall significance of these hybrid zones is that they give rise to selective breeding programs. They help us understand that individuals with desired traits can be bred together, and the offspring of those can then be bred together. And if we repeat this over many generations, we can produce our own variations. The ideal, the aim of hybrid vigor. And we've seen this before through artificial selection, where nature gives rise to new species in the thousands and millions of years. Humans, with attention to care and patience, can do it in 10 to 100. The truth is, speciation can occur rapidly or slowly and can result from changes in few or many genes. It really depends upon the organism and something known as the molecular clock. The molecular clock is a concept that identifies how likely mutations are to occur for a given organism and for a given region of their genome, based upon the polymerase error rate and its reliability. The overall evolutionary rate of an organism varies depending upon the organism and their behaviors. It's a complex equation, but to sum up, we can say that it really depends upon 3 primary factors. The first is the age of reproductive maturity. The second is the number of offspring they have. And the third is how many generations per year they're likely to achieve. Now, humans have very slow reproductive maturities. Even though we technically can reproduce at about 12, 13, most of us don't have offspring at this time. We wait until our 20s. We typically have 1 child per pregnancy. And because pregnancy takes 9 months, we typically have 1 generation per year. And even though we could have a pregnancy every year, Irish twins as they were, most have only 2 in their entire lifetime. And so overall, the evolutionary rate of humans is ridiculously slow. So 2 individuals, no matter how different they may appear, genetically, policy, are actually close to 99.9% genetically identical. Meanwhile, something like E. coli, which reproduces every 20 minutes, has 2 offspring, doubles every reproductive event, and can achieve millions of generations in a year, while 2 colonies of bacteria living side by side are roughly only 40% genetically identical. And so while evolution is occurring at every moment of our lives, in your lifespan, you will see more speciation events happen to other organisms than you will see occur to humans, because we are moving at a much slower rate. Our base estimates are that the interval between speciation events can range from 4,000 years in some cichlid fish to 40 million years in some beetles. And overall, we have a general average of 1 new species every 6.5 million years.